It's of course a pleasure to have Antonia Kaiser for this occasion, coming here. I'm sure many of you know Antonia, so I don't have to introduce lengthily. So Antonia, in fact, did a bachelor in psychology or in cognitive science in Germany, but then moved very quickly to brain imaging field for a PhD in Amsterdam. And from what I understand, she very quickly moved to a number of different techniques and also applications in psychiatry. She participated to a number of treatment studies, like the effects of exercise on anxiety, ADHD, a number of topics. I'm not sure it's an exhaustive list. And for a postdoc, she then moved to the EPFL in the lab of Li Jingqi, I never know exactly how to pronounce. Sorry. Where she became really more interested in spectroscopy. Still with this interest to link spectroscopy with psychiatry, clinical domains, and also cognition. And she's going to talk about that.
Thank you. Hi everyone. It's a pleasure to be here and to talk to you at the Breakfast and Science event. I'm going to repeat a few things that Patrik already said. Thank you so much for introducing me. Today I want to talk to you about from metabolites to cognition, advancing functional MR spectroscopy for brain function. So before I start, I wanted to go through who I am, actually more so that you understand what my background actually is and how I ended up where I am. I indeed started in a Bachelor of Cognitive Science in Osnabrück, Germany, a very small town. And back then, one thing that I was really missing in this bachelor was the application and the measurement of the brain, because we were really focused on computational methods to model the brain. So that's why I moved on to study cognitive neuroscience in Amsterdam, where we really had the chance to dive into more practical matters. And I did a lot of internships with a lot of measuring methods like EEG, but also methods to trigger the brain like TMS. And one of the things that I was exposed to was MRI. And that was at the academic hospital in Amsterdam, where they also, after my internship, actually offered me a PhD. And I focused on using multimodal MR methods to investigate cognition in the brain. And all of that happened under challenges to the brain. I'm going to dive into that in a second. After that, I had a bit of a year and a half of doing many things. One was a researcher position where I did more simulation work, and that was really in spectroscopy, and that really made me fall in love with this method, and also in the end actually made me apply to this job that I am in now. And on the side, I was also a lecturer in biopsychology, and that is a really multidisciplinary bachelor program, and I really taught a lot of lectures about you can basically say anything, and it would have been on the list. That then led me to be introduced to Li Jing, and we met online, and I think our visions of what we wanted to do in the future and what we needed from each other were perfectly aligned, and we decided to do this together. So I, right after my defense, a day after, moved to Switzerland and started my postdoc here. And that is what I'm going to talk to you about today. So one of the things that actually in my thesis really interested me, or was one of the conclusions that I could take from my PhD thesis, was that when you want to investigate brain function, it is quite difficult to just look at the resting brain, because as you can see here, a resting brain is firing in all kinds of patterns, and a lot of things are happening. It's a bit chaotic, and if you then actually want to find out what is functioning and what is dysfunctioning, you can quite easily get lost in this noise. So what I really thought and also saw in the studies that I ran in my PhD was that it becomes much easier if you give a trigger or a stimulus to the brain, and then you can actually see how the brain reacts to the stimulus, and through that you can see how the brain is functioning and can actually deal with this stimulus or with this trigger, or maybe also not. And if you then track whatever signal you can think of from the brain over a course of triggering and non-triggering, then you actually have a chance to see differences between people that might have some dysfunctioning or better functioning and less good functioning of that function in the brain. So I thought, okay, maybe it is better to probe instead of just observe, to really look at the brain under challenges and not just at baseline. And in the end, I really thought that functional MR spectroscopy was great for this, because we could then really track metabolic dynamics under these triggers and functions. And that is much closer to the underlying mechanisms of the brain. So that is exactly when I came here, and Li-Jing had a very great idea for a project already. So we wanted to together investigate functional MR spectroscopy as a sensitive tool for targeting neurometabolic alterations in psychiatric disorders. And actually, she had a much more concrete idea. She really wanted to do this in schizophrenia or also psychosis. So the clinical characteristics of schizophrenia can be roughly divided into 5 symptom categories. And of these 5 categories, only one can currently be quite effectively treated by existing medications and also treatments. But actually, one of the most devastating and interrupting symptom categories are the cognitive deficits, you can see here. And they include working memory, attention, and executive functioning. Welcome. In the past years, people have worked out quite a solid hypothesis on what underlying mechanisms might cause schizophrenia. And here you can see a schematic overview of these hypotheses. And from top to bottom, you can see the early to late neuronal changes, which also get more difficult to treat and also more difficult to reverse over time. And on the top, we see more metabolic mechanisms being out of balance, like mitochondrial function, energy metabolism, functioning of the glutamatergic system, and neuroinflammation. And these go hand in hand with dysfunctioning of the redox system and also oxidative stress. And on the bottom, we see more micro and macro circuit dysfunctioning, like executive and inhibitory system dysfunctioning and myelination decreases. And they lead to general dysynchronization of the networks, and then consequently to decreases in cognition down here. So the later symptoms have been investigated quite a lot in the past years, using more traditional MR methods, so like structural and also functional connectivity, using, for example, fMRI or DTI. And now we, as a group, we can actually investigate the more early symptoms, and that is using innovative methods for metabolic mapping. And for that, we use functional MR spectroscopy and functional MR spectroscopic imaging. And we hope that actually through that, we can find a sensitive tool so that we can target the neurometabolic alterations in schizophrenia in the end. So the rationale of the study is to search for mechanism-based, non-invasive early biomarkers for patient stratification to guide efficient monitoring of disease progression and treatment for diverse subgroups. And this hypothesis comes or is based on one pilot study that is actually being analyzed by one of our previous PhD students at the moment. And it's in a single voxel static spectroscopy study, which found that glutamate and GABA connectivity are disrupted in patients versus controls. And mainly when you look at the connectivity between the thalamus, so that is everything that says TH, and the prefrontal cortex, that everything is up here with the MPC. And you can quite clearly see that there are differences between these two groups. So one of the main batteries or lists of things that are being used to diagnose schizophrenia, and particularly for the cognitive symptoms, is a battery of loads of tests that target cognitive functioning. And one of the core cognitive domains impaired in schizophrenia or also in psychosis patients is processing speed. And processing speed is the ability to perceive a stimulus, consequently understand it and interpret it, and then to react to it in some kind of way. And it is declining in several conditions, for example, in aging, in psychiatric disorders, as just said, and also in neurodegenerative disorders. And one of the most important regions of the brain for this functioning is the dorsomedial anterior cingulate cortex. It's a mouthful. And it is indeed responsible for selecting actions, for divided attention, and for task switching. Therefore, we actually decided, okay, this is a great cognitive domain that we want to target and that we want to investigate further. And that's where I came in and set up my postdoc study. Investigations of metabolic dynamics during processing speed. So there are many tests out there that you can use to measure processing speed as a cognitive domain. And while searching for this, I came across loads of them, but one always stood out, and that was the Symbol Digit Measurement Task, SDMT. And that was especially because it's being used in MRI studies already and was mainly used with fMRI before. So there was already an implementation that works inside the MRI scanner. And our idea here was that we would implement this task in a block design. So you can see that there's always a rest condition, then a on condition, then a rest, and so on. And while the participant is in the scanner and doing this task, we can repeatedly measure MRS, spectroscopy, and that really enables us to then convert the spectra to concentrations of metabolites, here, for example, glutamate, and you can track it over time and hopefully see how it goes up and down depending on if it is needed or not in the brain. And then our plan was to compare these metabolic dynamics to cognitive and behavioral outcomes of processing speed. And here I wanted to quickly explain to you how this task exactly works. So here again, you see the block design. We have rest and we have stimulus conditions. And in the rest condition, you can see an empty table with a number below. I'm now going to start playing the video on the bottom so you can actually kind of see how this goes. So in the rest condition, that should be an easy condition. The participant is asked to just look at the number that appears below the table and read the number out loud. This number changes every 2 seconds, so it is not trivial, but at least it is as easy as it can be, and most people do not have a problem with this. Then we go to the active condition. Here you see the table is being filled and symbols appear below the table. Your task will now be to find the symbol in the table, match it to the number below, and then say the number out loud. So this also, again, sounds quite easy, but if you now look at the video, you will already see probably, maybe some more so than others, that it is actually quite hard, and that because it changes every 2 seconds, you get under a tiny bit of a time pressure. So the hypothesis and the goals of the study then were to characterize the lactate and the glutamate dynamics during this task. But not only do they go up and down, but also the latency and the magnitude of these increases or decreases. And doing that depending on the cognitive demand or effort, so the two conditions basically. And then 2, to investigate the relationship between task performance and the characteristics of these dynamics and metabolites. And 3, to compare the differences in lactate and glutamate dynamics in 2 populations that we used as an example for cognitive decline. So we decided to do this in aging, and we included young and old participants because the old participants should already show some cognitive decline. So we did that, and we went here to the 70 right next to us and set up the study. We used an S special sequence with a TR of 4 seconds and a voxel size of 20, 20, 25. Here you can see where we place the voxel, the yellow being the most overlap and the black being the least overlap between all 30 participants, all 40 participants we scanned, targeting the dorsomedial anterior cingulate cortex. And we were interested in two metabolites in particular, and you can see them here. I hope it's big enough. Ish. The upper being glutamate and the lower being lactate. And when you look at here this example spectra, you can appreciate that our data quality was actually really high, and we were actually able to look at these two metabolites. And additionally to MR scanning, we also did a big cognitive questionnaire and test battery to be able to see what the cognitive levels at baseline were of these participants. And in addition, with a collaboration with SCHUV together, we did some blood measurements. So here you can see the participant overview. Let me guide you through this. So we had quite a balanced sex sample with 50-50 being female and male. We indeed managed to scan young, around 26 years old participants and very old, around 75 years of age participants. They did not differ in education, in data quality, in the Stroop task, in a cognitive health questionnaire targeting mild cognitive impairment, and also not in psychological health. But they did differ in a paper version of a processing speed task and an executive functioning task, with the older people being lower than the younger people. We then went ahead and processed our MR spectroscopy data, and here I have a little bit of a side story for you. We, in the past year and a half or two even, developed a tool at the lab, the MR Spec Lab tool. I did that together with these two people and many more. That is a pipeline editor that really makes it easy for you to analyze any kind of spectroscopy data. And as an example, I used this tool already to analyze the data of the study, and included very standard processing steps. We fitted with LC model and used different averaging modes for the results that I'm going to show you in a second. And here is how the tool looks like. So you can really drag drop your data into this tool, your raw data for metabolites and also for water. Here you can see this pipeline editor where you can drag in some functions that are pre-coded by us, that are quite the standard functions that you would need, and really build a pipeline. And then by just simply clicking play, it will go through this pipeline and analyze the data and will spit out really nicely processed spectra for you in the end. If you're interested in this, this is our GitHub repository. You can download either executables or also the whole Python library. So I did that, and now I'm going to guide you through what I found. First, let's look at some behavioral results. Here on the left, you can see a plot of the reaction time as a population average in blue for the young participants and in orange for the old participants. And in gray, you can always see the on conditions, so the harder conditions, and in white, the easier ones. And if we focus just on the blue line for now, you can really appreciate that the young participants were quite fast in the rest conditions, so under 1 second to read out the numbers, and then really much slower for the hard condition, around 1.4 seconds. And when you then look at the old population, up here you see 1, it is much noisier, but 2 is also quite clearly in both conditions very much slower. So in both reading out the numbers and also in solving the task, they were quite a lot slower. And then here on the right, you can see the accuracy. So we could compare what they said and what they should have actually said. And you can see that the young people are at around 90 to 95% of accuracy. So also good nose healing effect. And the old people are quite a lot much lower, around 75% of accuracy. So this is all as expected.
So you record the voice?
Yes, yeah. We have a noise cancelling microphone in the scanner. Works really well, by the way. Yeah. So then here, a very busy slide, the results of the metabolites. Let me guide you through it. So here on the left, let's focus on this first column. On the top, you can see the results of glutamate, in the middle of lactate, and on the bottom of glutathione. On the top, or in this column, we can see a simple comparison of an average over all the rest conditions to an average over all the stimulus conditions. So just as a simple t-test, basically. And in blue, again, we see the young participants, and in orange, the old participants. In glutamate, we can see that the young increase to stimulus and the old decrease to stimulus. In lactate, we can see that both groups increase, but that the old in general, so also at baseline, have much higher levels of lactate. In glutathione, we can basically see that it doesn't change from rest to stimulus, but the young participants have higher levels of glutathione. So then the next step was to really resolve it in a time resolution that we can look at per block. So we calculated one average per paradigm block, and be aware that now we have a double axis. So here on the left, you can see the axis for the young participants, and on the right for the old participants. Otherwise, they would be very far away from each other, and the effects would be teeny tiny. So we put them on top of each other. But what you can appreciate is that as we actually see in these plots already, in glutamate, we can see that the young people really increase in the stimulus condition and decrease again in rest. And the old people do exactly the opposite. They increase in rest and decrease in the stimulus condition. In lactate, we see exactly the same as here too. It's the same pattern in both groups. They increase in stimulus conditions, decrease in rest. And in glutathione, you can see that it is a bit more difficult to make out patterns.
Can I ask you, what is the sample resolution? So how many number of averages do you have for each one?
It's every 4 seconds, and the block is 2 minutes long. Yeah. Yes, and then if you want to see something in this pattern, I guess what you could see is that in the young participants, it seems like there is some kind of fluctuation over the course of the experiment, but it might not be exactly locked to the paradigm setup, and it might be a tiny bit delayed over the course of the whole experiment. So then the third column, here we wanted to see, perfect, we see changes in these metabolites, and they do differ between the age groups, but do they actually have anything to do with the cognitive abilities of these participants? So we took the paper version of the processing speed task that we did before scanning, so the baseline values of cognitive ability here, and we correlated them with the change of the metabolites. So here you can see stimulus minus rest as an average over the whole package, and here the score of that processing speed task. And in glutamate, we see a positive correlation. So that means the higher the cognitive score, so the better they are in that task, the higher the change of glutamate. And in lactate, we see exactly the opposite. So the higher the cognitive score was, the lower the change in lactate is. And in glutathione, we see absolutely nothing. Very flat, nice line. So what does this mean? In baseline values, we saw that glutamate and glutathione were reduced in older participants, pointing to lower excitatory signaling and weaker antioxidant defenses. We saw that lactate was increased, suggesting the brain relying more on glycolytic energy production with aging, probably some kind of compensatory mechanism. In the change values, we show that we are able to capture them, that we are able to see the lactate and glutamate dynamics during a cognitive task, during a processing speed task, and we do see differences between young and old. And in glutamate, we saw that the young adults increased during stimulation. That is very much as expected. Many functional MRS studies before have already shown that glutamate increases with neuronal activity. In the older adults, we see a reduced glutamate response, and that could be just a different functioning of the aging brain. And that the stronger glutamate responses were linked to better baseline cognition, so better performers had higher glutamatergic signaling, which makes sense, the more efficient it is, basically. In lactate, we kind of saw the opposite. Both groups showed increases during the task and decreases during the rest conditions. Also, that has been shown before, that with neuronal activity, lactate goes up. And we see larger changes in older adults, which again points towards more of a compensatory mechanism. And higher lactate changes are associated with lower cognitive performance. So worse performers show more lactate involvement, probably needing to involve more lactate to actually get to the same performance. In glutathione, we don't really see a clear effect. There might be a possible delayed response, and that could be a stress-related antioxidant response. So the next steps, one, we also have resting state connectivity of fMRI scans that we can then calculate connectivity from. And I would really like to see if these changes in lactate and glutamate, but also the baseline values, are correlated with connectivity measures in networks of attention, executive functioning, but also just simply in the voxel location. And we're going to get results from the blood markers from our SHUV collaboration that we then can also correlate to the data. So one thing about the study is it is very nice, and we can see loads of things, and it is super interesting, but theoretically we, with these single voxel studies, always have to make a decision a priori about the location, right? So we made this decision to only measure in this one brain region, but what about the rest of the brain? So that's why one of the goals of the lab, the group, is to expand spatial resolution for this matter. And one of the first steps was to not only look at one voxel, but actually measure a whole slab, so a 2D acquisition, to be able to measure along this line throughout the brain. And that was being done by one of our PhD students, Shuwei, and she implemented a sequence so that you could do this in a time resolution of 30 seconds per slab, and a spatial resolution of 4.48, and then the slab is 20 millimeters thick. And as a first step to see if we can actually pick up signal with this, if it actually changes with a task, we implemented a very simple visual task. So meaning block design, rest conditions with just a black screen, and stimulus conditions with a flicking checkerboard. So very annoying stimulation for your visual cortex. We did that in 3 participants. They were processed with quite similarly to fMRI methods, so motion correction, co-registration to T1, but also MNI, brain masking, and some spatial smoothing. And then we did a general linear model analysis using NiLearn with a low frequency drift regressor, and FDR correction for 0.05 p-values. And here are the results. Here you see the 3 participants. As a reference, here in this column you can see the fMRI response to the same task. So quite classic, the occipital lobe, the visual cortex lighting up. And then on the right you can see glutamate, total creatine, and line width. Total creatine and line width being controls for what we're seeing. And in glutamate, you can appreciate that we actually do see an increase of signal in the occipital lobe with stimulation, and it is quite similar to where we expected it in the visual cortex. And then in total creatine and in line width, we do not see much. And then if you even look at voxels that are activated, you can really see the time course of glutamate changing. So black being the rest conditions and light gray the stimulus conditions, and the signal is really increasing by quite a lot. So you can see here the percentage change, which is around 6-7%. One other neurotransmitter that would be great to look at is GABA. But GABA is very difficult to measure, and that is because it is very low in concentration, but it is also overlapping with other signals in the spectra. You can see here in red the signal of GABA, and when you look at this black spectrum on top, you can really see that it is covered by lots of other things, and it is quite hard to resolve it and get that signal out. So because of that, there are very smart people out there like Andre who can program sequences that actually make it easier to measure GABA, and that is called editing. And he indeed did that, and he also did that for one slab through the brain, where you can, with 2 different pulses, stimulate different parts of the spectrum and therefore suppress certain signals. And when you here look at the black line, you can see that 2 peaks are actually resolved afterwards when you subtract them from each other. And we can see here quite a clear GABA peak, and here quite a clear glutamate peak. So he successfully implemented this, and he was able to do this in 3 minutes and 25 seconds per frame. And again, we wanted to see, does this work? So again, we implemented a visual task, but this time not only a simple stimulation versus rest paradigm, but also a so-called negative bolt visual paradigm, where you, instead of showing the full screen flicking checkerboard, you show a central checkerboard that then in fMRI shows a negative bolt signal in the center of the occipital lobe. And we did that because there's been one fMRS study. They placed the voxel exactly in this blue bit of the occipital lobe and looked at the metabolite dynamics. And they saw that GABA really played a big role in this. So therefore, we wanted to look at both of these. So here are the results. On the left, you can see the GLX, so glutamate and glutamine results. On the right, you can see the GABA. And the first column is simply full-on checkerboard, second column, the small checkerboard. So what do we see? One, in the small checkerboard, we can really appreciate that both in glutamate and also here in GABA, they're really in the periphery, so in the outer bits of the occipital lobe, positive signal changes. And then when you look at the full-on checkerboard, you see that these signal changes are really more in the center of it. Additionally, what we can also see in GABA mainly is some negative clusters around the periphery of the brain that are new. So what have we seen, or what have we learned? We've been able to implement a 2D fMRSI sequence to measure positive glutamate reactions to a visual stimulation task. We have a 2D edited fMRSI sequence that is a good tool for mapping excitatory and inhibitory metabolites and neurotransmitters during brain function. We have used experimental and also statistical mapping methods that can be translated from fMRI. And now with increasing advancements in spatial and also in temporal resolution, fMRSI holds promise as a non-invasive, metabolite-specific imaging technique for studying brain function in both health and disease. And that brings me to the last step and also my last slides today. And that is the application of all of this in an actual patient study. So recently we have gotten together with researchers from the HUG, Matthias Kirschner and his group. And we are going to hopefully soon set up a patient study together in which we want to implement both the fMRI methods and also the task that I presented in the single voxel study together in a patient population. For that end, we want to enroll 60 patients and 60 age and gender matched controls. And we are going to work together with his team. They have their own protocol, and we share as many sequences as we can. And we focus really on the metabolic mapping, and they focus more on fMRI-based tasks. So that gives us a multimodal magnetic resonance protocol, and ours is going to really focus on this glutamate and GABA connectivity mapping during a cognitive task. We're going to do the same with fMRI to have the comparison, and then hopefully use resting state fMRI to look at the connectivity measures. So what are the perspectives for the future or the next steps? We are still working on technical developments to improve temporal resolution for fMRI. Always faster is better. We are also trying to improve spatial resolution of fMRI. We already have some pilot data on 3D acquisition that gives us not only one slice, but actually 4 slices that covers quite a lot of the brain. Currently analyzing that also with a visual task. We're investigating and developing tailored statistical tools for fMRI, really borrowing from the fMRI world, seeing how does that translate. Do we need other regressors? What does that mean if the signal is much slower than the fMRI signal? But also maybe obtaining dynamic metabolic response functions, because the hemodynamic response function is just not applicable. So seeing if maybe with high temporal resolution we can aid that in the future and see what's happening on that regard. And then in the end really validate everything with larger samples and also apply it in this clinical study with a cognitive task. And that is all from me for today. I really thank everyone who is part of our group, the MET-MRS group of Lieging, everyone who's currently still there, everyone who has been there before, all the funding agencies and all the collaborators, and of course you for your attention.